big sky, big potential. In association with Mills and Reeve, this is Eastern Promise. Achieving more together. Greetings, dear friend, wherever you may be. This is the podcast that brings you the best, biggest and brightest from the east of England. And this week is no exception. So get ready for some true Eastern Promise. I'm Mike Rigby, and this week we're taking a special look at one of the most consequential research institutes in the east of England. The John Innes Centre on the Norwich Research Park is the beating heart of British plant and microbial science, nay, internationally. I'll be joining my Royal Norfolk Show co-host, Dr. Penny Hundleby, for a tour around her place of business. So join Penny and me as we go from bugs to barley and back again, with fish thrown in for good measure, on this week's Eastern Promise. The invitation for Eastern Promise to visit the John Innes Centre allowed me to finally tick off an ambition I've held since Eastern Promise first began 4 years ago. Not to be confused with the eponymous compost. In fact, the only rapidly decaying mulch here is this script. The John Innes Centre has been and will remain absolutely fundamental to not only plant and microbial science, but also to driving forward new legislation regarding the use of gene editing for plant breeding. And, as if that wasn't enough, the centre itself is about to undergo its next phase of evolution, the multi-million-pound HP3 project. We're particularly fortunate that our trip around the John Innes Centre is being led by a senior member of the JIC family, my Royal Norfolk Show co-host and podcasting pal, Dr. Penny Hundleby. Penny and I will begin at the very beginning, outside the John Innes Conference Centre, along with JIC comms guru, Hannah Arnold. This is, it would sound a bit weird if I said it was a dream come true, but it's certainly something I've been wanting to do probably since day one of Eastern Promise, and that is come to the John Innes Centre, the world famous, world renowned John Innes Centre. I was on a panel for the Chamber of Commerce in the forum, and they said, what's the real point of hope, of light, of, you know, where should we focus our, you know, efforts in raising up Norfolk and Norwich and get the economy going? And the first words out of my mouth were John Innes Centre. And it is an absolute delight to be here with my very good friend, friend of the show, as they say, Dr. Penny Hundleby. Penny, welcome. And we also have Hannah. Introduce yourself, please, so everybody knows who you are and what you do.
Thanks, Mike. I'm Hannah Arnold, Head of Media and Content at the John Innes Centre. It's lovely to meet you.
It's lovely to have you with us on Eastern Promise. Penny, welcome. You've been on the show before. Last year you came with me around the Norfolk Show, and again this year.
It's absolute delight to have you here, Mike. You know, I don't know, this is going to be sounding a bit of a mutual loving session here, but you know, we absolutely love all the work that you're doing, all the work you do to promoting us. And I think what I've really learned from following you on your podcast is just how diverse this region is, and you know, connections that you don't think are there to be made, and you're really good at sort of bringing everyone together. No, it's just, I've learned lots from listening to your podcast.
People will now learn about this tree, which is outside the John Innes Conference Centre, which many listeners will have been to, will have experienced, will have had brilliant events in there. I held the worst interview of my entire life with Professor Ben Garrett because I was totally starstruck and tongue-tied. And Ben, scientist important, yes.
The conference centre is where we sort of meet for big events. So we have the Friday seminar, but it's also, you can hire out the conference centre as well. So
yes, and I have in a previous life, but I don't think there's many people people, certainly in the Norfolk and the Norwich ecosystem and beyond, who hasn't been to an event at the John Innes Conference Centre.
And it's a great facility. So the actual main conference centre holds about 300 people.
Yeah.
And then we've got some really nice meeting rooms in there.
Concerts. I've been to a concert in there.
I've been to a wedding in there as well.
Really?
Yeah. So a former colleague, had her wedding in there, and I had a Kaylee in there. And, it was great.
Well, the atrium is a great place in itself, just to go in and mingle. And
yeah, so the Centrum's been I remember when the plans for the Centrum were first discussed, I remember thinking, I'm not going to walk all the way over there to have lunch. And it's so on our doorstep, and it's such a great opportunity to come and meet people from the other MBI institutes and the companies as well. It's that sort of real nice sort of chance collisions that people are always promoting.
For those who don't know what you do here and the very important work you do, tell us your role here.
Okay, so I've been at the John Innes Centre for over 30 years now, which I originally came to do a 4-year contract, and I remember my boss saying there's plans for this fancy new hospital to be built, but we'll be long gone by the time that's finished. And so I originally came working in genetic technologies, so I was a lab scientist developing protocols for genetically modifying, oilseed rape. And so I spent 30 years sort of doing that, and I've recently moved across to the Directorate as the Research Partnership and HP3 Hub Lead. So HP3 is our vision for the future, our healthy plants, healthy people, healthy planet.
Not The Prisoner of Azkaban, which is a joke I made last February, and it's still bad. But yes, it is so important. I've seen the absolutely magnificent vision, and it looks amazing. You must be, I hope you're going through the blueprints, the architectural drawings, and say, Penny's office, we'll just take that wall out, make it bigger.
Yeah, I think no.
Very responsible, I like it.
There has been so much in the planning behind this. So we're now we've now sort of started the building work. It's going to be a phased, the building's going to work, it's going to take over 5 years. So we're hoping to be fully open and operational by 2031. But the planning behind that, it's been over like 7 years of discussions and architects. There's been a lot going behind the scenes just to make sure that what we're building is going to be sustainable and future-proof. To take us through to the next phase of our journey.
So, that's it's basically it is to take the amazing plant science. Well, plant science doesn't do it justice, but the amazing work you do at the John Innes Centre, and basically taking it on to the next level, giving you that scope to do so much more than you ever dreamed possible, I suppose. And it's just, quantum leap, not to mix my metaphors totally, but of plant science into the future.
It's huge.
We're
delighted. So we have, we've gained UKRI infrastructure funding. So a sizable chunk of investment is coming our way. So just short of £320 million from the government. We're also in a fundraising phase to get another £80 million. So if you have a few £1,000s down the back of your sofa, get in touch. In total, over £400 million investment into new buildings and infrastructure for us. And so we'll take you into the courtyard. I'm going to show you the existing buildings, talk a little bit about our history and how we've evolved over time, and and where we hope to go in the future.
Well, to just close that thought, I think that's cheap at twice the price. Ooh, now this is nice. You talked to me earlier about the pond, and here we are. Good God!
Top pond, and we've got 2 sturgeon in here. So Jules and Nicola. The one that's called Nicola is because we had a previous staff member called Nicola Sturgeon, so it obviously lended itself quite well.
So no, they were, not after the former First Minister of Scotland, Nicola Sturgeon.
No. So and then we've got these guys here, and then down here, amazing science going on here, and we're showing you the pond.
I should describe. There's a pond with sort of lilies and. There's a waterfall into another area, but there are 2 massive fish just lazily ambling around,
Yeah.
There is a little science link here, though, Mike, if you are interested.
Please do, Hannah, because otherwise I'm just sort of...
Well, I only found this out a few weeks ago, but actually our scientists, along with partners at the UEA, have done some amazing work on the algal blooms in the Broads, the Norfolk Broads that we all know and love, and, you know, obviously one of our sort of flagship attractions in the east. We actually did a lot of work working with anglers on the Broads to try and look at how to support the health of the Norfolk Broads and, you know, predict when the algal blooms were going to happen, how to solve them. And the UEA has actually recently just taken up that research again and has got some funding, so it's really exciting. So some of our work does impact the natural environment in the east of England as well.
Well, it's another one of those things where you wouldn't think it, but then when you learn about it, it suddenly makes absolute sense.
Absolutely.
Why, you know, of course, the John Innes Centre are involved in tackling these things.
For sure.
And keeping our waterways healthy, because, as you rightly say, Hannah, the tourism is a massive part of the Norfolk economy, and it dwarfs other parts of the region. Yeah. But wow, that is a lot of... Is it koi? Is it just koi? Well, I'm, you know, learning about my fish here, but...
Yeah, if you want to...
There's a lot of them.
Pop your finger in the water, they will come and suckle your finger.
I will pass. I will pass. Oh, that sounds like...
And so we've got our heron decoys, because we have little visitors.
You do have two mighty-looking heron
you can just
decoys.
hear. So this is the air ambulance that's going over. So we're located next to the Norfolk and Norwich University Hospital. I can talk about the park in a second. What's up, please?
But yeah, I mean, many listeners will be familiar with it, but for those who aren't, it's a huge co-location. The proximity is what makes it really exciting, in that it is literally 5 minutes over the road to get to the hospital and all the facilities therein.
So I thought if I stand you here, Mike, I can show you some of our buildings that we've got on here, but also just sort of talk about how the John Innes has evolved over time. So we get our name. So the original name John Innes comes from Mr. John Innes. So he was actually a property developer and philanthropist, but he had a real passion for garden cities and thought that we needed to be living in green space for our sort of, you know, well-being.
Oh my goodness!
And so when he died, and it's reported that a week before he died, he changed his will. And said, I want to, you know, give, I think it was £380,000, equivalent of about £51 million in today's money, to establish a horticultural institute to do a bit of a mixture of discovery science but also applied science. Here we go, the helicopter's going back over again.
It's circling, looking for a landing.
We've timed that very well. So the long shot is that they then formed this charitable trust, and that still exists today in the form of the John Innes Foundation. And so they built this horticultural institute, and its first director was a guy called William Bateson. And he is famously reported as coining the term genetics. So up until 1905, when he famously writes this letter to a Cambridge professor, there was no word to describe the study of inheritance. And so he said, if one was to coin such a word, genetics might be a good word. And, you know, that is in everyone's sort of vocabulary, isn't it, that word genetics? So I think we can very much lay the claim that we are the bedrock of plant genetics, and that encompasses so much of what we do. And so he was the first director, and he bought a lot of the learnings of Gregor Mendel. I don't know if you know Gregor Mendel. It's sort of the history of sort of genetics and study of inheritance. So he sort of brought that learnings to the UK, and it was very controversial at the time. So he kind of sort of embedded that. So this building we know as the Bateson building, after this guy. And when the John Innes Institute, as it was, moved from its original birthplace in Surrey to Norwich, and that helicopter's going again.
It is. There's a plant around the corner from where we live that does ready meals for everyone from Aldi to Marks and Spencer's. And every week the air ambulance seems to be landing outside the plant, and I keep thinking, somebody really needs... Well, I just think, I hope they're checking the packs of sausages, and there's, you know, why is someone got a ring on it?
Oh no.
the John Innes Institute moved to Norwich in the 1960s, and it purposely came to align with the newly formed, at the time, University of East Anglia. So that had opened just a few years prior. And I think this goes back to, I think it was Zuckerman's report, which said that research institutes do really well when they're aligned to universities. It's that mutual talent pool.
Yeah.
Also, at that point, we had the Institute of Food Research, which had just opened across the road. And so very much it was that sort of magnet for bringing, so we had food research, and then we had the sort of plant genetics that John Innes bought, and with the university. And then in those early days, we had joint partnerships with the university. So we had associate, professors who came to sort of do, to really sort of strengthen our genetics and applied genetics. And then we sort of managed to act. So we're very much acting as this magnet for bringing sort of like-minded research to sort of strengthen the kind of work that we do. So then we sort of, we drew up the virus research unit from Cambridge, who came to sort of join us on site. We had Streptomyces research from Glasgow University. So we're both plant and microbial research that goes on here at the Research Park, and we've actually got the largest density of plant and microbial scientists anywhere in the world. So definitely in the UK. I'd like to say globally, but I'm pretty sure we are
definitely up there, if not the top.
Yeah.
Wow.
And you just don't realise that this is happening on our doorstep.
Well, this is one of the reasons I'm so keen. I've been always so keen to get it, because the scope of what you do, as you say, touches so many areas.
Yeah.
And if you go around the Royal Norfolk Show or the Suffolk Show or any kind of something in there will have had at some point. I'm pretty confident. I don't know this, but I'm pretty confident that the John Innes Centre will have been involved at somewhere in its life cycle.
And what I find fascinating is people who've come sort of studying, Streptomyces. So this is bacteria that lives in the soil. When you pick up soil and you smell it, that smell is sort of products that this bacteria is releasing. And if you think that plants and microbes, they don't have that ability to get up and run away when they're in danger, so they're producing these products to protect themselves, and we're able to now harness the sort of those products that they're releasing, which has benefits for biostimulants, biopesticides, but also for medicines as well. So a lot of our antibiotics that we get are now, secondary metabolites from bacteria.
And the science came from here.
Yeah, so actually some of that early engineering biology was a result of the Streptomyces work. So understanding recombinant DNA technology, being able to engineer plasmids to produce different proteins, that's all sort of stemmed from there.
I mean, it's such an important thing, because I remember seeing a presentation not that long ago that, I think it was in a weird context, but it was basically saying that pretty soon you won't be able to grow grapes in the Champagne region of France because it'll be too hot.
Yep.
However, here in the UK, and I bring that up because of a sort of fundamental part of what you've been doing here at the John Innes Centre is looking at the science behind these things, taking things like climate change and looking at how do we adapt the plants that are so fundamental to human life, to animal life, to all life, and make them resilient in the conditions that are to come. And I feel guilty like pouting on Hannah now, that who's dealing with far more important things than this podcast. But I mean, that must give you as a comms professional such a rich tapestry of stories to draw on, to put out and say what this centre does is of such fundamental importance. You have to come and see.
It's almost too much. It's incredible, the amount of amazing work going on. It's sort of blown my mind over the last half a year that I've been here. We mentioned Gregor Mendel earlier. One of the big papers we had about a week ago was from our germplasm resource unit, which you're actually going to see later on, and they have finally unlocked some of the mysteries of Mendel and his original genetics.
So what we're standing in now is the link building, and so just going back to our history and timeline for the John Innes Centre. So this building here is known as the Biffin building. So
there is a very, very impressive resin, is it bronze? I don't know. Statue of Sir Roland Biffin, FRS, 1874 to 1949, if you want to Google it. With a very, very fine moustache and floppy hat.
And do you know what?
Every bit the country scientist.
He would have originally, when he was built or sculptured, was holding a pipe.
Yes,
But obviously with our anti-smoking policy and healthiness, his pipe has been replaced with some wheat, some wheat plants there.
Do you know what? It didn't even occur to me. I just thought
he was
'twas ever thus, you know. He was ever holding the wheat, thinking of, yes, well, about this wheat.
So he was the former director of the Plant Breeding Institute in Cambridge, and so when PBI was privatised, their research and development section moved here. So the non-commercial side moved here, and that brought alongside the researchers have really sort of strengthened our wheat work, our barley work, our oilseed work, and our cell and developmental biology. So that kind of was these guys. So they moved over late 80s, early 90s. So that was them moving over. So that was the Plant Breeding Institute, and we knew it for ages as the Cambridge Laboratory until we renamed it.
I did see that on the door.
Which confuses a lot of people.
Which does, thinking, am I lost? You know,
yeah. So that's the history of why they're here. And then over the road, you can just about make over. So alongside our conference centre, we have the Chat Building. So named the Chat Building.
Chat Building.
So that's named after Joseph Chat.
It's not what you do there. It's named after Joseph Chat.
Sometimes.
And so it was the old nitrogen fixation laboratories, which came from the University of Sussex. So that's when they came over, and they came over here in 1995, which was also the same year that I joined the John Innes Centre. And it was the bringing together of those three different entities, so the John Innes Institute, the Plant Breeding Institute, and the Nitrogen Fixation Laboratory. In 1995, we became the John Innes Centre.
Right.
And so that is kind of the sort of foundations for that.
What I got from that was a lot of these, this institute, this laboratory, this organisation came here, which is exactly what you want to hear. This is exactly what we want to get across, that this is a magnet for scientists, for experts in the field, including yourself and Penny and Hannah here. So where next? Because this is already blowing my mind.
One extra building that I need to tell you about. And so we have on this wing here, we've got the Sainsbury Laboratory.
Yes.
So the Sainsbury Laboratory is a separate entity as such. We're government funded, so we get our funding, a lot of our funding, not solely, but mainly from BBSRC and UK.
Which is one of the research councils.
And the Sainsbury Laboratory is Gatsby funded, and they've got very much a focus on plant health, and we have a lot of shared institute strategic programmes together. And so one of the things that we're doing within our £400 million new investment is that we are building a new main laboratory which will house for the first time the John Innes Centre, those three different buildings, but also the Sainsbury Laboratory. So we're going to be under one roof, brings this critical mass of expertise under one roof, which is really exciting. But it also brings together all our technology platforms. So we have around about 14 to 16 technology platforms, and what's really great about those is these are our technical specialists, and we'll go around and look at some of them. So we have things like our bioimaging platform, our bioinformatics platform, our crop transformation platform, proteomics, metabolomics, lots of omics.
Yes, we said earlier that we would put a glossary at the end for anyone who wants to know about the omics.
But what is really a real pull to come and work with us, because we have these fantastic technology platforms and specialists, and these are the real core memory of the institute, and so a lot of our grants our funding will tend to be sort of 3-4 year programs, and so we get a lot of early career researchers who come in, and for them to be able to upskill and learn all of this would be pretty much impossible, but now they can come in and tap into, so I want to do a bit of research, we've got a platform that will extract DNA for them, we've got a platform that can do this and that for them, and it's something that is very unique to research institutes, and one of the sort of selling points as to why we do so well compared to the universities.
Well, yeah, absolutely, because, we had a fairly recent episode of the podcast where, on this very park at the Quadrum, Zampler, who are a Cambridge plant materials company, came to Quadrum basically to make use of their artificial colon.
Yeah.
So just to make sure their probiotics actually got to where they needed to be, and weren't lost along the way, as it were.
But we've got another really shining example of that is you'd have heard of tropic bioscience.
Yes.
So I think so. Not only do we have the different research institutes. So on the Norwich Research Park, you've obviously got John Innes, the Sainsbury Laboratory. We've got the Quadrum Institute, which you're a big fan of, the Quadrum. The Earlham Institute, the university, the
Earlham's
hospital.
on my to-do list if you're listening.
Okay. So yeah, we could definitely link with Nick Hall and his team over there. So we've got all of those institutes, but we're also this amazing ecosystem where there's over 40 companies on the park, and one of them is Tropic Bioscience. They came to the park to talk to us as consultants back in the day, and they wanted to set up this new company, and they wanted to know what are the bits of kit. So that laminar flow that you just saw being wheeled through. So they're the bits of kits that company would need to be fully operational. And I think it might have been John Clark who said, well, why don't you just set up shop here, make use of our facilities and infrastructure, so you can pretty much, hit the ground running. And so they came here 8 years ago as a company of about 6 or 8. They now employ over 150 people. So it's a huge success story. They're using gene editing and precision breeding to work in bananas, coffee and rice. But they're also a mutual pool. So actually a lot of the people that come and train here will then go on to work within some of the companies that are on the park. So I think it is this beautiful sort of ecosystem of working together.
Yeah. And it's all about, we talked about, when people come and people go, but it's about, I think one of the most important things, and I know you agree, is creating those ambassadors, those, who have such fond memories of working there, that they may come back in the future, but they go on and take those connections and say, actually, you need to speak to my former colleague Penny, who is still at the John Innes, and she can tell you all about X, Y, and Z, and HP3, which is, there's a board about it just over there, which is of such huge importance. Sorry, Hannah's taking a photo slightly.
But we also talked about this over coffee, that we've had so many recent visits from government departments who've come on, and actually a lot of the people working within these government departments are former alumni of ours. And so, we really do have this, this ability to not only develop the scientists of the future, but the people who are going to be sort of, impacting policy and on all those kind of areas.
So my name's Jake Richardson. I'm the manager of the Bioimaging platform at John Innes Centre.
Well, this is magnificent, because if I was to be crude about it, I'd say that this kind of looks like if you were to if you had a coffee machine, but the coffee was made in a bucket. It kind of looks a bit like it's like Mr. Spock's microscope. It's a fantastically scientific looking. Well, it would be, wouldn't it? Piece of kit, but Penny, you are absolutely right. It looks nothing like your traditional microscope.
It's the size of a small car.
It's the size of a small car.
How much would that have cost?
It's probably bigger than my wife's car.
I mean, we've done a few upgrades over the years, and the initial cost was around 1.2 million. And then since then we've had around the same amount in upgrades to cameras and things, and so the initial outset was this was originally purchased. We had one before this, but this allowed us to do cryo-TEM, which was kind of an emerging technique and something that a lot of John Innes Centre wanted to get involved with, and that's when I turned up on site and started. It was about 7 years ago, and then since then, with the kind of technique picking up and more groups using it, we've then been able to apply and get funding to be able to improve the microscope. And so that was with a new camera, and that's got a much higher resolution capability. So we can see more,
we're not
essentially.
Only going into the cells, but you're going further down, and now we've got the technology where we can actually almost image at the atomic level, which is kind of your cryo-EM.
Yeah, so we're getting into kind of 3 angstrom resolution, and so we're able to look at the kind of structures of proteins.
Define for me an angstrom, if you would.
So you've got 1,000 microns in a millimetre, and then you've got there's tenfold that angstrom, so it's 10,000 angstroms to a millimetre. And so we can get down to a kind of around a 3 angstrom resolution. So it's very small. So it's essentially, and it depends on the technique. So we can't do that for everything, because depending on how we've prepared the sample, that's a huge, and it depends on the technique. So we can't do that for everything, because depending on how we've prepared the sample, that's a huge kind of limitation of how resolved we can get. And traditionally, material scientists, they've got samples that can, you can see gold atoms. And so when we're doing alignments on the microscope, we'll use actually material sciences kind of samples because they're much, better to work with. The tricky thing with plants is that there's lots of kind of open spaces in them and they're not very dense material, and so we can't get much stain in there. There's, we're firing electrons at a sample, so those electrons can generate charge and energy themselves. They have radiation damage, and so although we're seeing it at very high resolution, we have to be careful because it's not impervious to damage. It will be getting damaged all the time as we're looking at it. So we have to either be very quick or very slow and careful. And so there's lots of kind of, it's not even just like a seesaw of like give and take in one thing. There's so many different variables that you have to consider.
So it's so far removed from the age of peer in and focus, and twiddle the thing. And
getting the black marks on your eyes.
Yes.
Yet we still have many different light microscopes. We've got far more light microscopes than we do electron microscopes. And so we've got 3 electron microscopes, 1 TEM that you've just seen, which is the transmission electron microscope, 2 scanning electron microscopes, which we shorten to SEM. And then the rest are, in the rest of this kind of corner of the building are all light microscopes. So we have microscopes that are similar to the ones that you're kind of mentioning at school, all the way to ones that use lasers. So that we can get the highest resolution achievable using light. And that allows us to use living samples often.
So one of the things that we do through our platforms is, so imagine you have an early career researcher or a PhD student who wants to use a little bit of this to further their research. And so they can either come and work with Jake as collaborative partners, or Jake could do the work for him, or they can come and learn how to do it here. And so we've got, you just wouldn't be able to do that kind of research on your own, and having the technology platforms really helps unlock other people's research and take it to that next level. So one of the technology platforms we're going to see is our genotyping platform. So again, as a service provider, rather than having lots of individual scientists extracting their own DNA from plant tissue that they're working on, we've got a centralised unit. So we just send in boxes of the various plant tissue we want sampling, and the team here will do all the DNA extractions for us. So this is all complete change. So is this our new massive CER room? So this is the one that we're talking about, which is probably going to be one of the largest CERs that we have available.
No, this is for seed storage. This is kind of halfway between the gene bank and the field.
So it's for researchers just storing the stuff that they're, continually working on.
Mr. Fothergill need not apply.
You can if you want. So we're working on various crops, and then they're growing in the field station, which is about 4 or 5 miles away.
Yep.
And then you often start processing the samples as they come out of there. So, for example, this is all like counting seeds and looking at the dimensions of the seed, the size of the seeds, width and length, and things like that. And then over there's bits of equipment for like near-infrared reflectance, where you look at the chemical composition of the seeds, the amount of protein in them, for example. Wheat, for example, gets exposed to the rain just before harvest, and that can reduce the quality of the grain, and that's measured in something called Hagberg falling number. So you're measuring things like that here: Hagberg falling number, grain, protein.
Hagberg falling.
Yeah. Iron and zinc, so nutritional components of the grain, things like that. We do something called XRF here for those measurements. So it's a post-harvest seed processing lab, and that's where you get a lot of the interesting traits for selection for better varieties.
We touched upon fibre as we were walking around.
We did indeed.
And you had some really good stuff that you presented at Governing Council.
Oh, yeah. Fibre's a nice story. So, so increasingly you get the health benefits of fibre. It's much more than keeping you regular, right? Mm-hmm. It's a gut signalling molecule. So when the microbiome interacts with the fibre in your gut, it produces short-chain fatty acids, and then they act as a signalling molecule to increase how full you feel, so satiety, change hormones so that it reduces the risk of diabetes and cardiovascular disease. Huge health benefits.
So what's your best source of fibre?
Well, that's a great question, because the best thing, for example, for whole grain, for wheat would be for everyone to eat whole grain. but they don't do that. And there's a long-term trend of people, in spite of, I think if you went around the general public and said, you know, what's better for you, whole grain bread or white bread, most people would know whole grain.
Yeah.
But they don't buy it, you know, and sales of whole grain bread never go up. And in fact, if people eat bread, it's white sliced, you know, especially in lower socioeconomic groups, which is where the health problems are. So the strategy evolved over the last probably 30 years in the UK, where geneticists and nutritionists start to say, okay, well, let's improve the white bread. And it turns out you can increase the fibre content of white bread. It's through a molecule called arabinoxylan. And actually, it's have no negative effects. If you eat it, you'd never know the difference. And you can increase the concentration of arabinoxylan with quite simple genetics. The way you measure fibre actually in here would be through viscosity. So you'd mix the flour up with some water and you'd see that with the higher fibre levels, it's just a little bit more viscous. But by the time you've baked it or made some products out of it...
So why I mean, this may be sort of out with the opinion of the John Innes Centre corporately, but is it just a case that we haven't been able to do that at scale yet, but we will soon, or is it just because we're still...
No, it's a much more interesting problem than that. And actually, I think we thought we understood, and we had meetings with the economics department at UEA, the Centre for Competition, and there are actually quite complex economic reasons why things like that don't feed through for public good. The technical phrase is coordination failure within the supply chain. So, you know, if you think about it, wheat starts off with breeders crossing it to make new varieties. Farmers grow it, you know, under the advice of agronomists, and then the farmers are working to something called the recommended list, which is basically the shopping list of what the UK wants out of wheat. And on that list, it says yield and disease resistance, resistance to lodging. It doesn't say anything about nutritional benefit.
We've had conversations with breeders and they're going, well, actually there's no money in that, so the money is from making yield. So it almost needs some sort of government policy.
I was just thinking that exact thing, that if you were West Streeting, you would say, because of the prevention, it's very hard to sell Treasury on prevention because it's upfront cost and there's nothing to show for it. You're paying to ensure an absence of something. That down the line, these things will be not eliminated, but certainly vastly reduced, and with it the expense on the exchequer.
It's exactly so. If you look at salt reduction,
yeah.
So I don't know if you noticed, there's less salt in bread than there was 15 years ago, and that was done. I think it was a bit of push and pull from intervention from the government, and the hard intervention, along with the sugar tax and things, and then the industry saying, okay, we'll do everything we can to try to reduce salt. And they did. There's much less salt in UK wheat-based products than there were 15 years ago.
Yeah. That
was really done by the millers and the bakers.
Yeah.
And the consequence of that is 40,000 people a year staying alive, right?
Well, that's pretty good.
Exactly. And then for fibre, the health benefits of increased fibre are much greater than the benefits of reducing salt. So it can happen, and I'm still not absolutely sure of that's why we're talking to economists, because we really want to understand the mechanisms by which it happens. The genetics is done, totally deliverable.
But can you imagine if we could get this high fibre white wheat, you know, and we coupled it with these vitamin D enriched tomatoes to get pizzas? I mean, look, pizza is used by so many people. And I know we've talked at length about children with additional needs, and I've got an incredibly fussy eater child, and actually trying to get nutrients into children with special education needs. This product would be fantastic for us.
It would be. And you're right, I think it requires intervention. Even if it's just... I mean, I'm not given to bursts of Trumpian economic thought, but there is something to say that, okay, I mean, I've always wondered why we can't do things like... even if it's an award that, you know, it's the award of Sweet Fanny Adams, but just to hang a gong on someone and say, we like these people, they are doing things of which we approve, and we are going to give them the quality mark of government thinks you're lovely, bang. Yeah. And that was, well, we want the quality mark of government thinks you're lovely. Well, don't sue to get it, show us you can do it. And
well, on this one, we delivered the genetics, but the strategy and the nutrition and biochemistry came from Rothamsted.
Yeah.
Rothamsted's big medal at the moment is 96 job losses and 25% cuts. You know, the area of nutrition and genetics and nutrition is not really functioning well within the academic sector at the moment. One thing we're doing is just saying to the funders, we'd be really interested in ways of bringing that back into the centre of what we do. It's one of the main deliveries we could have.
And I think one of the things that was interesting that always used to be, I confess I don't know whether it's currently being done, and it would be wrong to say it isn't, but I remember when a minister would straddle departments as a kind of unification. They would be DEFRA and DH at the Department of Health. They would be Department for Science and Technology and et cetera, et cetera. So I think that's the kind of thing we need more of, those kind of...
There is no department for nutrition, right? What department is it split between? It's a bit of DEFRA, a bit of Food Standards Agency, a bit of Net Zero, Department for Net Zero, and that's one of the points that came across from the discussions we've had is when things are going to work in government, somehow it's got to converge on a track, and then they're saying, yeah, we're going to do it.
Sounds like a job for the John Innes alumni.
So this is what we were talking about, that actually quite a lot of our alumni are in government department positions, and actually having people go into those jobs that have this broad range of science knowledge to actually how, you know, science and policy need to be much more hand in hand.
So, well, this is the Insectary, as I was saying, this is our containment facility, which means that it's designed to prevent the insects we have from escaping.
Right.
It has two sections. This one here that we call low containment, where we have native species or exotic species like these ones. But if they escape, there's no problem because they won't be able to become established. In this case, it's leaf-cutter ants. They're from the tropics. They won't survive the winter. And that's assuming that the queen will escape. The queen is very happy there, and the rest are workers that will die without reproducing. So.
They look very, very happy and very busy with their occasional bits of leaf wandering around this kind of labyrinthine tiny little cave-like system in this tank of ants.
Yeah, that cable-like system that you see is actually that they are farming.
Yeah.
So there's a fungus that they grow.
Oh.
And the fungus produces little bodies that they feed the larvae with that. So basically they're farming mushrooms.
I see.
And they have been doing this for 30 million years or so.
And there's one with a very impressive bit of leaf. He's scuttling across ant cam. I love it. I'm going to get a photo of the ant cam with ant.
So actually they don't eat the leaves. What they do is that they bring the leaves to the nest, they chop in little pieces, and they feed the fungus that. And what they eat is the fungus afterwards.
So they're feeding the fungus?
Yeah, so basically they are cultivating the fungus with the leaves that they cut.
What a symbiotic little system, wow.
Yeah, and it's even more interesting, the system. So the interest in terms of research here is a microbiological one. So there's a group here that's led by Matt Hutchins. What they're doing is that they're interested in the antibiotics that the ants produce, but not the ants actually. It's a more complicated system. So the ants have a symbiotic relationship with the bacteria that they grow on their bodies. Sorry.
Yep.
and so they grow on these chest plates here.
Oh, right.
Yeah, and they produce an antibiotic that they use to treat. It's an antifungal, and they use to treat the nest, so they don't get any other parasitic fungus that will invade the culture, basically.
Wow.
So then these antibiotics are quite good, apparently, and they're very interested in trying to identify them. And perhaps in the future they will be useful for humans to because they have found that the bacteria have a hard time to become resistant to those ones. So then it could be.
So you were quite right, Penny. It is far more than I first thought. Incredible.
So the antibiotics of the future may be a result of this little roaming ant.
Well, yeah, but we don't know, do we? Look, do we?
Yeah. Yeah. So that's one aspect of that. But then another big aspect is, as Penny mentioned, is insects or invertebrates in general as pests of our crops. And Sam will talk a bit more about the mechanistics behind that, how molecularly these things happen, this interaction between the plants, the insects, and the pathogens that that somehow, in many occasions, are transmitted by the insects.
Yeah, I mean, as you said, when you came in, pollination is something that people think about when they think about plants interacting with insects, but there's a huge problem, and you probably know from your garden there's all sorts of insects that are pests.
Yes.
Like the aphids, the green flies, and that's something we're interested in.
Right.
And they not only chew on or suck the sap out of your plants, they transmit viruses. So most plant viruses are actually moved from plant to plant by aphids, and that's where they're a real problem. And we've relied on using insecticides to control them in agriculture for the last 50, 60 years, really, and it's pretty clear that that's not the solution for the future. So we're trying to find alternative ways to deal with that. If we can use plant genetics to get plants or crops that are resistant to aphids, then we don't need to use pesticides.
At all, no.
And then we're not going to be harming the pollinators and all of the other beneficial insects.
Exactly.
We've kind of mentioned it before about that untapped genetic diversity, and this is one of the really nice examples of where we've gone back to some of those historic varieties to find ones that are resistant to slug attack.
Right. Slug attack.
So basically the Watkins collection is a collection of land races of wheat that were collected before the Green Revolution kind of diminished the genetic diversity in crops a lot. So there are all these varieties that were collected from many different countries. Were supposedly adapted to local conditions. And there are a treasure trove of genetic variation that you can use to screen for things like resistant to slugs. So then but this is a project that started also just to get an idea of the timescale we are talking here. So research is slow compared to so it takes time. We're talking about this project that started kind of in 2015, right? It's screening some of these land races, not all of them, because there are about over 1,000.
So what's land race? Just
a land race is basically a variety that's adapted to a territory.
Right, got it. Makes sense when you say it like that.
One of the really neat things about this approach, because I talked about our reliance on insecticides, which we've been using for 50, 60 years or longer, the crop breeders haven't had to worry about breeding crops that are resistant to insects, whereas they have put a lot of focus into yield and disease resistance and other traits.
Right.
These Watkins lines, they predate chemical insecticides. So we might, in modern crop varieties, we may well have lost a lot of natural insect resistance that might have been present in historic collections. So by going back to these historic collections, we might find useful traits, including insect resistance, that have been lost from modern crop varieties.
So yes, before we go in, just here you have a small panel where we show what we have. And
yes.
So most of them are self-feeding insects. I think, Sam, you're going to be talking mostly about...
This is my favourite.
The peach potato aphid.
Yes. Despite its name, it's actually not a very fussy eater. And
right, exactly.
Most insects, you'll find most herbivorous insects will have a favourite plant, and they'll be a specialist on a particular plant. But a small minority are real generalists, and they can feed off a huge variety. And Miser's persicae, you'll find it on 500 different species of plants. And that makes it a big pest as well, because of course it can move from the hedgerows on the field margins into the crops and spread diseases between the two, whereas the specialists don't do that. And other pests, similarly, like the whiteflies, not so much the glasshouse whitefly, but the tobacco whitefly Bemisia, this is a generalist pest, also a sapsucker, and just has a very varied diet and moves from plant to plant.
Do you default to the Latin just out of interest, or do you sort of move between the two?
Yes, because people have different common names for insects. The peach potato aphid is also known as the green peach aphid, is also known as the peach aphid by different people, and so.
But the Latin name is universal.
Yes.
Got it. As I say, if the common name is an indicator of what it eats, then I'd be a bit terrified of meeting the bird cherry oat aphid.
Yes.
Or even was the glasshouse whitefly. So.
Yes, but most of these, like aphids, are generically known as greenfly or blackfly, which doesn't really represent the diversity, because there are 5,000 odd different species of aphid around the world.
Almost as many as mammals.
Yes. Yeah. Well, the rate of discovery is supposed to be going up exponentially, isn't it? The rate of discovery, particularly of beetles.
Yes, and wasps.
And wasps. Oh. Yeah.
Should we go into the high containment lab?
Let's go. I will show you that. There are a few others that these are particularly interested here. Can you leave the bag here?
Yeah, sure. How far are we going?
Because we need to put the lab code. So
Okay, yeah.
We're coming back out this way.
No, as long as we don't go too far.
Yeah,
The
you want to leave
recorders.
in this office, you can.
I can take a recorder. It'll be fine.
Do you need it to be?
No, it'll be fine. Because we can always use it off the actual. Right. Whoa.
So you have to go here, and this is part of the containment measures. So you need to. Can I grab two? This is our left codes. What size are you?
Oh, big. Portly.
Look at that.
Portly.
This small. What size are you?
Large. There's a large.
This is large.
Yeah.
Are you small there?
Medium.
Medium better. So.
You sure?
Yeah.
Have you been in
Sam,
that?
you have your own.
I loved it.
Okay.
You have your own.
I do.
Mike wanted to know about your zombie beer if you get time.
Right. Okay. Noted. Yeah.
Right, we'll grab mine because mine is here.
I got in as an early adopter, and I got one of the nice, convenient, head-heighted.
Yes, the early adopters always.
Well, this is cozy.
It is, isn't it?
So these lobbies is to prevent the insects from escaping as part of the measure.
Yeah.
So it's all dark. The only light is red because most insects cannot see red. So this is all pitch black for them.
Are you able to just...
Oh, sorry.
That's all right.
I forgot about it.
No, it's good. That's the point. You forget about it. It's good. You get great stuff.
So basically, if any insects from there, there's the part that we call high containment, things that we don't really want to escape, come here, they will see all this will be black, except dark, except that. And they will go there and then bang, right. So,
Well, it's nice and clean, so obviously doesn't get much in the way of work.
well, we have to monitor them as part of the requirements for the license from DEFRA. We need to monitor all the straps to ensure that they haven't escaped.
So you've got to have the paperwork from DEFRA. So.
And I don't think it will be of interest for you, but then we have also strict regulations in terms of how we deal with the waste. We have to freeze everything, then we have to clean with a washer at a certain temperature, and then everything that leaves this facility is also autoclaved afterwards.
Right, so frozen then burned.
So the principle is anything that comes alive, but only humans live alive.
Mostly, usually.
So we keep each colony of insects enclosed in a cage like this. And here we've got Mysis persicae. This is the green peach aphid. And
yes.
you can just about see, there's a few specks. You can see that dead leaf, which is covered in some alive aphids. So they reproduce incredibly quickly. I don't know if you know about their reproduction, but aphids have a fantastic life cycle. They're mostly asexual. So they give birth, their population is completely female. They give birth to live young, like us, not like most insects that lay eggs.
Really?
The live young are clones of the mother, genetically identical, made from unfertilized embryos. So they're identical. They're born, because they don't need to have sex, they're born pregnant with embryos that are also pregnant, of themselves. Which means they can reproduce incredibly fast. And that's why when the weather is getting warmer like it is now outside, you'll see these colonies of aphids appear and grow incredibly quickly, and that's what makes them such powerful crop pests, because the populations can just explode, which is a fascinating study. It also makes them very easy to work with, because you can grow a population very, very quickly and get a lot of them. And then they'll just sit there quite happily sucking the sap out of the plant. And we're really interested in, because plants have immune systems like us, and they are able to defend themselves against attack, recognize when they're under attack. But the aphids are able to circumvent those plant defenses, the immunity, the toxic chemicals they make, and the responses and the changes in gene expression. And they do that all through proteins in their saliva that they secrete into the plant when they're feeding, much like a mosquito when it's feeding on you will, introduce painkillers and stop your feeding and stop your blood clotting. They're also introducing factors that are preventing the plant responding. And we're trying to understand those molecular mechanisms. And
so it's a bit like it's all right, nothing to see here.
Yeah, exactly. They're stopping the plants, and so we're hoping by understanding that we can change the plant genetics and restore the plant's ability to mount an effective defence against those aphids. And exactly, yes.
Is the pool open yet? Just sorry, no.
No,
I love the pools. You shouldn't show me that.
I wanted to show you the pool.
I was talking to
But, so I think as part of the John Innes Foundation, having a social component was really quite important to them. So this is kind of sort of a legacy. So we've got some amazing facilities in there. We've got the squash courts. We've got the big sort of, you can go and play basketball in there. And I don't know when it's going to be open. I think the cover's still on there. But did I take you in there last time?
You've shown us, yeah. You showed Sally and myself. You must sign in and out at reception before swimming.
We'll just go for a little walk through, just because. So on a Friday, the bar's open. You can get your John Innes beer in there as well.
Oh my God! You make it sound like keeping up appearances in Hyacinth Bouquet. Large lab swimming pool, room for a pose.
but it's great. I mean, so these are over 30 years old. I mean, I've been here 30 years, and this was all here. But we've got a squash court in here.
Squash court.
Can't even, don't even know where the lights are. That's how long ago I was last in here.
Well, I can see the squash courts.
Yeah, you can sort of see it in there. Squash court in there. There's the viewing platform up there. We've got, the pool, and this is just such a lovely, actually it's pretty much gearing ready to be open. So this opens between May to September. And actually, it was built to a lot of our surplus heat goes into heating this. So there's like a reason behind it. But we have barbecues here in the summer, and then the rec centre. I'd have to send you through the men's toilets separately if you go that way.
That's okay.
We'll go this way to the other end.
Well, if you're listening and you're interested in a career at the John Innes Centre, why wouldn't you be?
And then hopefully Mike will appear as if by magic.
We appear in several. This is nice.
On a Friday night, the bar's open, so it's a subsidised bar, and everyone just sort of comes together. And you think that we've got people from all around the world. So actually, building that sort of social side for them is really important. I think, sort of for
it's really nice.
For well-being
and cosy.
It's a real community. I'm quite new here, and I felt welcomed from day one.
Yeah. And actually, the big sort of summer parties that we have here, some of them are quite legendary. There are still, I'll never know whether that really happened, but there's, reports of, people diving into the pool at the end of a party as well. You
are painting a very wild picture of plant scientists, I have to say.
And then we've got, badminton courts. And again, way back in the day, they used to put on this sort of almost like a panto type sort of production to bring all the staff together. I think that kind of sort of, died a death. But, I think when we were much smaller, it was easier. But, we've kind of got bigger. So it's trying to still create those opportunities for people to come together. And I think sometimes when you come together socially, that's where, again, those sort of chance conversations happen, and that's where, a lot of connections and collaborations come.
Yes. And partners use this as well. It's not just John Innes Centre staff, it's open to everybody.
Oh, right. Well, there you go, even better.
Yeah, so you can bring your family for a swim, and you have to bring your daughter sometime. We'll do it as an official.
Don't. She goes every Wednesday, goes swimming.
My huge thanks, as ever, to Penny. I'm hugely indebted to her and her support for Eastern Promise, which has meant a great deal to me personally. And I'm thrilled to have returned the favour with a much-anticipated tour behind the scenes at the JIC. My thanks also to everyone we saw on that day. There was much more recorded that I can fit into our usual length. Later this year, I'll be bringing you my interview with the Centre's new director, Christabel Wowie. And I want to find out what you want to know. So send your questions for Christabel to me in the comments below via DM or to podcast@easternpromise.org.uk. And that's all there is for episode 143 of the Eastern Promise podcast. I have been Mike Rigby and will be so again next week when we bring you the highlights of Cambridge Tech Week. As I write these words in early August, I still haven't the faintest idea what will happen. How exciting! That's all still to come in just 7 little days. So, until then, bye for now. to hear other episodes of the Eastern Promise podcast and to find out more about what we do, go to our website at easternpromise.org.uk. Eastern Promise is a Pudding Shed production in proud association with Mills and Reeve, achieving more together.